Welcome to this ReadStore tutorial where we want to combine the ReadStore database for NGS datasets with Nextflow, one of the most powerful tools for orchestrating bioinformatics workflows. And for those bioinformaticians here that are not so familiar with Nextflow, we will first have a look at some basic concepts, go through some of the training resources where you can find more information and educate yourself on the specifics of setting up Nextflow workflows. And then the second part, we will actually see a Nextflow workflow, which is designed to pull data sets directly from our ReadStore databases so that we can integrate both together and use the power of ReadStore to actually manage your data while using Nextflow to execute and build bioinformatics workflows on top of this. So let's get started. We're here in the Nextflow training resources. And this is the best place to start, obviously, if you want to educate yourself on Nextflow. There's a number of really helpful tutorials. You can start with the Hello Nextflow section here, which is explaining all the basic concepts here and a number of examples. And then there's a fundamentals training, which is also a great tutorial to get started. And here in the basic concept section, we will have a look at some of the essential components of Nextflow, and those are the processes and the channels. And the processes are the building blocks that hold individual bioinformatics steps of our pipeline. And the channels are basically queues that are linking different processes and make sure that the files and data sets that need to be processed in our bioinformatics pipelines are coordinated between the processes. And here's a nice example that should give you some overview here. So a channel can hold different data sets or different chunks of data or different files or whatever we need to process as input. And then the channels are connected to individual processes. And within a process, every element from a channel can be analyzed. So for instance, if we have a channel that holds a number of FASTQ files and we have a process that is performing a QC step, then the power will be that we can design a flexible workflow where the QC process is reading from a channel and then performing the QC step on each element of this channel. And those two components are the basic building blocks of a Nextflow workflow later. And if we want to go down a little bit, there are some of the other great advantages of using Nextflow. And this is particular in the number of supported platforms here, the flexible languages that we can use to define our workflows, and also the orchestration tools. So Nextflow is really good in chaining software that is living in Docker containers, which is super helpful for cleanly separating dependencies for individual bioinformatics steps. There's support for a number of cloud platforms and HPC environments. So if you're interested in scaling your bioinformatics on your AWS or on your Google Cloud platform, there's a lot of connections here. And if you have some specific cases, please feel free to shoot me an email if you have questions about how to orchestrate and scale bioinformatics pipelines. Happy to support you there. So then we can go to where we want to start, and this is the Nextflow for RNA-Seq tutorial. And this is a workflow that is designed to process RNA-Seq data and perform an alignment and certain QC steps. And we can start in the part one. And part one of this tutorial is actually related to defining the individual bioinformatics steps and then seeing how those can be executed in Docker containers. And the steps that are executed here are a FastQC step. This is meant for checking the quality of the sequencing data. A trim galore step, which is designed to cut the sequencing adapters and perform a QC step, then again on the trimmed data set. An alignment step using the HiSat 2 software, which is a tool for performing alignments of sequencing reads against the genome. And finally, a multi-QC step, which is also a reporting tool for collecting QC statistics. And those four tools are now checked in this part one of this tutorial. We see that what is done here is they use containers that contain the dependencies and definition of the tools. And then using Docker, you can start a container, and in this Docker container, you can then execute the individual bioinformatics tools. So for instance, here, FastQC has been run in a Docker container. There are different steps here. As I said in the beginning, we have a FastQC step. The next one would be a trim galore step, which is clipping off the sequencing adapters. And in the part 2, this is where the actual alignment is performed, so where the sequencing reads are aligned to reference genome. What you need for this is first an alignment index that is always dependent on which software you use for alignments. And here, the HiSat 2 software is being used. And for this, first a genome index is created, and this genome index is then used in the second part to perform the actual alignment of our FASTQ files of the individual reads against this new index. And finally, there's a multi-QC container defined that is pulled from container registry. And in this container, the multi-QC software is executed, which is a nice tool to combine different reports and combine sequencing metrics. So what we're actually interested in is the part 2 of this tutorial. We have seen how to define the individual bioinformatics steps in Docker containers in the first part. But now what we want to look at is how to chain those in an actual Nextflow workflow. And for this, we look at the part 2. And the first thing that comes up here is a workflow definition. And the workflow is defined in this .nf files, and this Nextflow files. And here is an empty template. So workflow usually contains either the processes or include statements that import modules that hold the individual processes. There's a section for pipeline parameters. We need to, for instance, define the inputs to our workflow, and then the workflow itself, which is creating the input channels and then chaining the different processes. So this one is empty now, and in the next steps, we want to start to fill up this process for this RNA-Seq pipeline. And the first step is the QC metrics. So we want to run FastQC on an input FASTQ file. And for this, there is a module that's being created. So a module is a part of a workflow, as I said, that can be imported. And in this module, we define this FastQC step process. So to define a process, this is relatively simple. There are a couple of statements here, which we'll quickly walk through, and this process definition, and that makes up one of the individual bioinformatics steps that can then be chained in the workflow. And the first statement here is a container statement, and these container statements define a Docker image, which should be used to execute the process commands in. There's a published here, which is copying the results later into an output folder that is basically at the position where the Nextflow pipeline is invoked. Then we have, very importantly, an input statement, which is taking on the parameters that are required to actually run this process. And there are output statements that define what comes out of this process. And the script path is then so the script part is then defining the actual instruction for the code that we want to run in the container. And here it's a bash command to run the FastQC, and the parameter here is the input path to the reads variable. So it's running FastQC on a path that points to a FastQ file usually. And FastQC is producing two kinds of outputs, which are a zip file and an HTML report, and those are defined here as outputs. And here this instruction is basically saying, take the path that I put in, take the simple name, which is the name without the file ending, and then add the FastQC zip or HTML endings and emit this under these variable names so that they can be used in the next process. So input is your a FastQ path, output are two paths for the result files of FastQC, and that makes up the first process. Now, in the next step, we need to import this process into the workflow file. For this, there's an include statement where we want to define the name of this process. And then we add a parameter here in this point 1.3. And the parameter for this workflow that is named reads, which we also need in the first processing step, is then the path for a FASTQ file. And going into the workflow definition itself, we need to set up a channel. And a channel is here an input channel from a file path. And we take the parameter that we defined that contains the path to a FASTQ file, and then creates a channel from this path. And this channel can then be used as input for the FastQC process here. And our workflow now consists of two steps, which is saying create a channel from an input path to a FASTQ file, and then this channel is taken into the FastQC process and executed there. And then in the next step, what is done here is just a short test run. So Nextflow run is the command to invoke this workflow, and that should run FastQC on the FastQ file. Now, in the next step, we want to add the adapter trimming and post-trimming quality control. And for this, there's another module that's being defined here. So here's another module that's been created, the trim galore module, and a process is defined that is called trim galore. Again, defined with a container that should actually run the code, a publish directory to hold the results. So they're now in the trimming subdirectory. The input, again, is the path to FASTQ file. And then there are three outputs this time, which is the trim FASTQ file, a report, and then again, two reports for the FastQC output that is run on the trim FASTQ file. And the command here that is being executed is simply trim galore, and then including the FastQC, and then the path is pointing to the input reads file as in the first place. Now, same story again. This process is being imported in the main workflow file, and in the main workflow file, there's step added here in the workflow. The process is added. And this process now takes in the same channel as the FastQC process. So the Nextflow run will then execute this in a way that both FastQC and trim galore actually run on the same input channel, and this is what's been tested here. So finally, there's of course the alignment step, which is at the core of the RNA-Seq pipeline, and for this, there's a new module that's being created that is making use of HiSat 2, the software that is used for the alignments. And here you can see that the process becomes even a little bit more complex here. So the HiSat 2 align process now is actually running on a different container. So you can already see that this is a container that contains probably HiSat 2 and some tools. And again, the publish dir is adapted so that the outputs of this process are written to the align subdirectory here. And inputs are now two, again, the reads, and which is a path to a FASTQ file, a trimmed FASTQ file in this case, and the path of an index zip file. And this index file contains the genome index that is required for performing the alignments. The script that's executed here is also a bit more complex. So this is now bash script that is executed then inside this HiSat 2 container. And first, what is done is that the index zip file is unarchived, and in the second step, the HiSat 2 command is executed. To import this process into the workflow, it's the same as before. So the include statement includes the HiSat 2 align process into the main workflow file, and we need to add another parameter here, which is the path to the genome index. We've seen in part 1 that there was a step where a genome index was created using HiSat 2, and then the HiSat 2 process align is added here also into the actual workflow. And we can now see here that it's a bit more complex here because the input channels to this process are actually coming from another process. So the trim galore trimmed reads output, which is the path to the trimmed FASTQ files from the trim galore step, are now the input channel to HiSat 2 align. And we have the file which is pointing to the zipped index file, which is the second input here. And again, one of the powers of Nextflow is that it's actually now figuring out how to execute this. So there's obviously a dependency here, so that trim galore needs to be executed before the HiSat2 alignment step can be executed. And that's actually the job of Nextflow to run this. So again, a test run here, and that makes up our RNA-Seq pipeline. And what we'll see in the next part is how we can build a similar pipeline where we are not taking the input files from a parameter, but where we pull them from the ReadStore database. So in the second part of this tutorial, we want to look how to integrate the ReadStore database, which we can use to manage our NGS projects, together with Nextflow to build a workflow that pulls the data directly from the database so that we don't have to manage or mess with file path at all. And for this, we now logged into the ReadStore app from the browser. We can move to the datasets overview and see that we have 3 example datasets here, which are RNA-Seq data from tumor and normal in replicates. And those we want to now access from a Nextflow workflow. So we now opened a Nextflow workflow that we're going to use to make the ReadStore API calls from within a Nextflow process. And for this, we start by defining a process that is dedicated to extracting the file path for a given ReadStore dataset from the ReadStore database using the ReadStore command line interface. And we can quickly validate that this works. So if we say here in the terminal ReadStore dataset list, we should validate that we have a connection to our ReadStore database and that we get all the datasets that are stored here. And this is also true. So in our parameter section in the Nextflow workflow definition, we have now a parameter that's called ReadStore dataset, and that is the name of the dataset as it is stored in the ReadStore database. So this enables us to access datasets independent of the file path so that we don't need to mess with this anymore. We have 2 more parameters, which are an output directory and the path to a Salmon index. So for this pipeline, we actually use Callisto for performing the transcript quantifications. But coming back to the ReadStore API calls, we have here a process that is called getReadStoreFastQ. And as input, it's taking the name of a dataset here that we defined, and the output is a tuple of 2 path to read 1 and read 2 of a given data set here. And in the script, so in the bash script here, we define the API call, and the API call in this case is ReadStore dataset get -- name, and this will return -- read 1 path, and this will return the path of the read 1 FASTQ file that is associated with this dataset here, and then return this together with the path of the read 2 file. And this enables us to get the file path from the ReadStore database by providing simply the dataset name as an input parameter. The next step in this workflow is, as we've seen before, a FastQC step. So here the input is again the name of the dataset and the path. And then the FastQC command is executed here, and the output is a folder that is created for this specific dataset, and the input are the reads here. Again, we also specify here a container to run FastQC within this container and then publish the outputs to a folder that is defined by the outDir parameter that we set before. And third step here, third process is then the quantification step. Also, this one is running in a container, and the input here is, besides the dataset name, the path of the Salmon index file, the path to the reads. And what is happening here is that Salmon quant is called, and this is using the Salmon tool in order to perform a transcript quantification from the FASTQ files using the index that is provided here. And the output here again is simply the folder of where the Salmon outputs are written to that is named by the dataset. And the last step, and this again goes back to the ReadStore database, what we want to do is that we actually store the transcript counts that we get from Salmon back in our ReadStore database so that we can access the file path for all our bioinformatic pipeline outputs again through ReadStore so that we don't have to mess with this with a file path anymore, and we can access everything through the dataset name. And for this, we have this upload quant results process here, where we perform an API call to the ReadStore database, which is ReadStore Pro Data Upload. So we upload process data. This is what the Pro Data command is doing for a data set with a name that we defined in the beginning. We give the data set a name that we want to upload, and we call this TX, so transcript quantification. The -T is the type of the data set, and we call this here Salmon, and then the path to the transcript counts, which in this case is this quant.sf file, which we want to register in our ReadStore database. And finally, we chain all these commands using the workflow statement. So first we extract the reads tuple using the get ReadStore FASTQ command. We perform the quantification. The input to the quantification command is now the name of the FASTQ data of the ReadStore data set, the path to the index file, and the channel that contains the reads that come from the ReadStore database. Then we perform the FastQC step as well on the channel of the reads. And finally, we upload the quantification results and take as input here the path to the directory that contains the Salmon outputs as well as the dataset name. And this is now a workflow which enables us to run an RNA-Seq pipeline on top of the ReadStore database without us having to actually manage dedicated path to individual files, but we can manage everything through the dataset names, and we could even extend this workflow now so that we can run this on whole projects or specify dedicated workflows based on certain metadata or something similar. We will now run this actually. For this, we just say Nextflow run and give it the name of the script. And this can take a second. And we can see now that it took us roughly 3 minutes to execute this workflow, and all these individual processes succeeded. And if we go back into our ReadStore app, we can actually navigate to the process data page here. And what we should see now here is that we have a new file, and that is called TX, transcript quantification. And this is what we upload through the Nextflow workflow. So that was it for building Nextflow workflows in combination with the ReadStore database. I hope you liked this. If you have questions or comments or need help with setting up Nextflow or ReadStore, please reach out. And otherwise, I wish you a great day.